Our next speaker, Dr. Patricia Hunt, is the Meyer Distinguished Professor at the School of Molecular Bioscience at Washington State University. In 2003, Dr. Hunt discovered the adverse effects of BPA, a plastic compound found in many products like plastic water bottles, on the reproductive systems of mammals. She continues to work on the impact of toxic chemicals in plastic products, including BPA replacements. Please welcome Dr. Patricia Hunt.
Thank you, and I'd like to thank Rick and Heather for inviting me to be part of this. It's a really louder. All right. So I don't think about the structural aspects of these chemicals. I think about their biological aspects. So I'm a biologist, and about 20 years ago, I put my foot in the plastic sea, and my life has never been the same. So I'm going to try and tell you a little bit about that journey and the mission that it planted me on, and where I think we need to go. And I'm going to try to do it quickly so we can have questions. So I work with animals, and I know for a lot of you think animal research is a terrible thing. I think it's an absolute necessity, and I'm going to try and show you why. Because this is the burning question that's driven my research career for, a few years now. We as a species don't do a very good job of making eggs. We make a lot of mistakes. And if you actually look at the number of mistakes we make and you plot it against maternal age, you see a really bad thing, that this is the incidence of chromosomally abnormal pregnancies like Down syndrome, plotted against maternal age, and by the time a woman's in her mid-30s, you can see that this curve's going off the charts. So what I want to know is why? What changes with age? What's really interesting is there's a little blip down here that's real too in the teenage years. So we make a lot of mistakes, half of the species anyway. And my question has always been, why? It's not a good recipe for success as a species. So many years ago, 20 plus, I decided that what we were seeing in our human eggs was suggesting that maybe what was going on is subtle changes in our hormones with age were influencing this process and leading to these mistakes. Okay, that's really hard to study in us, so I decided we should be studying this in mice. Mice don't make nearly the mistakes, but they're a great model for the process. And we were in the course of these studies, and wow, we were getting data suggesting, you're on the right track. When all of a sudden, everything in my lab fell apart, and some of you have heard this story before, because we had a temporary worker who came into our mouse room, and he was told, this is the floor detergent and this is the cage washer detergent, and he picked up the floor detergent, used it on the cage washer, and he damaged all of the plastic cages and water bottles used in our facility. They were all made of polycarbonate plastic, which is comprised of polymerized BPA. And so all of a sudden, my animals were being exposed to this chemical. And all of a sudden, all my normal control animals started to show me wacko eggs like this one. Okay, what you're looking at is a green spindle and red chromosomes. The chromosomes should all line up at the middle, nice and tidy, so that they can segregate. And they're all over the place so that they would give you chromosomally abnormal eggs. Okay, well, this was disaster. We couldn't do any experiments, but it was also riveting because it suggested not only were we on the right track, that maybe this was hormone-like things that were changing, hormone changes that were giving us chromosomally abnormal eggs, but this was a chemical that has hormone-like properties. Okay, BPA or bisphenol A is what we call an endocrine disruptor because it interferes with our body's hormones. So all of a sudden, I found myself talking to everybody, talking to reporters, talking to clinicians, talking to legislators, talking to parent support groups about these chemicals and about whether or not we should be concerned about them. And I realized one thing. Scientists really suck at talking to the general public. We're terrible. really terrible. You know, if I'd had a chemistry professor like Mark, maybe I would know a little bit of chemistry, but you know. And so this made me realize that the most important thing that I could do is teach the next generation of scientists to be better at communicating. Because let's face it, your taxpayer dollars pay for our research. But we never feel compelled to tell you what it is we're doing with your money. And some of what we're doing with your money is really, really important. So I've been on this vendetta to try and make them understand that they have to talk to people. It's their job. When I ask them, think about your favorite teacher. Imagine that teacher. How many favorite teachers can you think of? Most people can only get one or two. When I ask them what made that teacher really, really good, it takes them a few minutes to get around to it. They finally go, they were enthusiastic. They made the complex really simple. They were funny. Okay, I go to a lot of meetings, and I would not say that that characterizes most of the scientists who stand up and talk. Okay, but I have a really complicated story, and I needed to simplify, and I didn't find this easy. I'm interested in making an egg. It takes a long time to make an egg. It will not surprise you to know that women are precocious. Okay? We start this process when we're developing in our mother's womb. All of the eggs are going to ovulate, start their journey in your mother's womb. And a really important part of the process happens there, and then the process arrests, and a little bit later, these little developing eggs get packaged into follicles. And then when this female becomes sexually mature, which means that her brain and her gonads start talking to each other through hormone signals, we start to recruit follicles to grow, and hopefully that growth culminates in the ovulation of a single egg every month. But that single egg doesn't just take a month to grow, it takes more like 3 to grow. So this whole process is super, super long. What we were doing when we tumbled upon this BPA effect is we were studying this part of the process, the end of the process when the egg is completing its growth and is just ready to be ovulated. And we saw this effect of BPA exposure. Well, in the intervening 20 years, we've looked back here when we don't think there are any hormone signals that are involved in the process, and we see an effect there too. And other people have looked here, and we see an effect there too. So we see an increase in chromosomally abnormal eggs if we expose mice here. We see changes that will lead to an increase in chromosomally abnormal eggs when that female grows up. And we don't quite know what this part of the process does, but it changes things in a way that does not look favorable. So we need mice to study this, because I can't study this in humans very well. I just can't get volunteers to donate their babies for my research. But that part of the process, that fetal part's critical. Because what we're doing here is we're shuffling the deck. Okay, you got 2 chromosomes. For every chromosome, you've got one from your mom and one from your dad. The name of the game, whether you're making eggs or you're making sperm, is to pass on 1 chromosome. Not just dads, not just moms. So there's a bit of wild magic that occurs because they find each other. The DNA gets broken, and it gets repaired as these little anchors that hold these chromosomes together. And that gives us genetic diversity, which may not mean much to you, but it's survival of our species. Okay, so these little ties that bind these chromosomes together, these breaks and these rejoining between your mother's and your father's chromosome, they're critical. And what we were finding is exposure to this chemical, this bisphenol A, was changing that process in a bad way that increased the likelihood that we were going to get mistakes when these females became mature. Okay, so my first BPA problem resulted in a whole bunch of news reporters getting really interested, because it's like this chemical we're all exposed to, if humans behave like mice would mean an increase in miscarriages, an increase in chromosomally abnormal babies like Down syndrome. This one was really much more significant, but I couldn't really explain it to reporters well because all of the eggs you're going to ovulate were starting their journey here, and with a simple exposure in mouse for a couple of days, I could affect all the eggs that developing fetus was going to ovulate as an adult. How the heck do you study that in humans? You don't very well. But here's what we know after 20 years. We know that this change not only occurs in mice, we can induce it in monkeys too. Okay, I never really wanted to do mouse research, but it's a critical tool for geneticists. I would never work on anything higher than a mouse until I had this big problem. And I had a colleague who said, you know, Pat, I work on monkeys and we should do this study because we need to know whether we're going to be affected the same way. And this is the closest thing we can get to that, answering that question. Surprisingly enough, my colleagues who work on this little nematode, this little soil worm, see exactly the same thing when they expose their worms to this chemical bisphenol A. So what's really wild is this effect of this chemical is so conserved among species that are so different. The other thing that's really wild is this. This is what I call a grand maternal effect. It's a 3-generation hit at once. We're exposing mom. It's affecting that developing fetus and can alter a lot of things: brain, heart development, lung development, development of the reproductive tract. But if that is a female fetus, it's affecting that developing ovary and changing the likelihood that those grandchildren will be born, will be normal. So we did this monkey study, and I wasn't, really totally comfortable with this because, these are monkeys. They're so close to us. We only had 6 monkeys that we were exposing and control animals to. But I said, wow, if we're going to do this study, then we have a whole lot of colleagues who work on different aspects of this chemical and whether or not it affects the developing organ system. So we got on the phone to them and we said, would you take the tissues from these monkeys and tell us if you see effects? So we had someone who studies the brain, someone who studies the lung. The mammary gland is very, very sensitive, the lungs, and the uterus. Okay, I did my studies and I looked at my animals and I ranked them, who was most affected and who was least affected. And I said, hey guys, would you rank your monkeys too? And I got the brain and went, wow, there's going to be hot monkeys here who are really affected and ones that are less affected. And then I got the study for the mammary gland and I went, And the heart. Mammary gland and heart are kind of similar. And then the lungs. Wow. And the uterus. Lungs and uterus? And I scratched my head and I looked at this and I went, what does this mean? And I realized what it meant is every single monkey was affected, but to different degrees in different organ systems. But every single monkey in at least 2 organ systems ended up at the most affected top 2 slots. This is exactly what you would see if you fed something toxic to humans, like bisphenols. Okay, so that's the female. What about the male? Okay, we were really interested in that because we had seen this effect in females, and we said, well, let's look at the comparable effect in males. Okay, if you read the popular press, you've been hearing sperm counts are falling, right? It's been all over the news, and it's thought that maybe this is due to exposures, like estrogenic chemicals. So when we did our studies, we're back in mice again. What we found is that, yes, we could induce effects with exposures, and it gave us a mechanism whereby we would drop sperm counts. because we were exposing right after birth in the male, and we were seeing changes that would knock cells out in the process of making sperm and drop their sperm counts. No one ever leaves my talk singing and dancing, you know, it's just not a happy thing. So what about humans? You know, this means for me, I don't really have questions whether or not this is harmful to us. I'm more concerned about how we can prevent exposures. But we kept getting from manufacturers the idea that, you know, that it doesn't matter. You can ingest this stuff and you metabolize it very quickly and you eliminate it. Gone. Boom. So that's true. If you swallow it, first pass through the liver, it's conjugated into two different forms. You either glucuronate it or you sulfonate it. If I was a chemist, I would know what that meant. The predominant form is this form. But if you take it in another way, through your skin, you're all going, why would I do that? Okay, so next time you pick up that pressure printed receipt, they say, do you want your receipt? Just say no. Because when you pick up that receipt, it has a matrix lined with BPA or some other bisphenol, and you take it in through your skin. Extra bonus points if you wear hand lotion or you've just eaten greasy French fries because you take in more. Okay, so how you take it in makes a difference in how long it lasts. When you ingest it, yes, boom, it's eliminated. When you take it in through your skin, the bioactive form is around longer. So this is the form we worry about. We think these forms are inert, but there's a little bit of suggestion, maybe not so much. So contamination is a huge issue for us, not just in my mouse room, but when we collect biospecimens. If I got blood from every single one of you, I would have to worry about the needles I was using to draw blood, the tubes I was drawing blood in, the materials in the laboratory in which we analyze the samples, because all of those can give us contamination. We did studies with monkeys, another set of monkeys and a whole group of people. We looked in monkeys, we could feed them radio-labeled BPA. So we could ask, how long does it hang around? And we could see, it depends on the way we delivered it. If we gave it to them orally, boom, a big spike in 1 hour, drop. When we got the same animals pregnant and repeated the experiment, boom, a little blip. Where'd the rest go? Yeah, you guessed it. Cross the placenta to the fetus, and we could actually see it there too. We could actually see it in the fetal ovary. So we've been interested in human pregnancies. We collected a lot of samples, maternal blood, cord blood. We wanted to know how exposed are human fetal tissues, human fetuses. Well, you know, at the same time, BPA is getting kind of a bad rap, you know? You guys know you don't want to buy it. You want to buy BPA free, right? You'll pay more for it. Because we went to state legislators and we said, we want this stuff out of baby products. You know, we want it out. And they must have laughed. They said, okay, we'll take it out. BPA free is a whole bunch of other bisphenols. Through the magic of chemistry, we can tweak this molecule into not one BPA, but 50 plus bisphenols. And the way our regulatory system works, the onus is on the feds to say it's not safe, not on the manufacturers to say before it goes into production and use that it is safe. Okay. So I have a lot of questions going on in my lab. I'm really interested in a lot of things. And then all of a sudden, we run into the same thing that happened 20 years ago, which is we're in the middle of our experiments. We're excited about what we're finding. And all of a sudden, we don't have controls because we are contaminated again. My first cages, as I told you, were polycarbonate plastic. You just touch them with this high pH detergent and they degrade and start leaching bisphenol A. Well, I'm not stupid, so we switched polymers to polysulfone, which is a more stable polymer. But when we saw this shift in data, we started to notice that these cages were starting to show us some hazy little signs of maybe damage. So we put some alcohol in those cages to extract whatever was coming off, and we sent it to our favorite chemist. And he came back and he said, Pat, it's bisphenol S, which is one of these replacement bisphenols. So I got on the phone to the manufacturer and I said, we have these polysulfone cages and they're leaching bisphenol S. And they said, not possible. There's no BPS there. Because this is polysulfone, which is polymerized bisphenol A and another bisphenol diphenylsulfone. So I thought, well, what we're going to get is we're going to break bonds and we're going to get bisphenol A, and we're going to get this diphenylsulfone. So this is what we need to look at. But my chemist, who's a wonderful chemist, Roy Girona from UCSF, said, no, Pat, you can't generate that because you're going to break that ring and you're not going to get that. However, if you just move that break site over, gosh, I'm so challenged, you would get bisphenol S. So, we can know what it's made of, but we don't necessarily know what it's going to break down into. I live over the mountains on the Palouse. When I show this slide, people think it's the biggest golf course in the world. It's not. It's the biggest wheat growing region. But this is my ray of hope. Don't blink. So we had all these contaminated mice again. What we did the first time is we got rid of all the cages, we got rid of all of the mice, we started all over again. This time we knew that if we exposed animals and we saw these changes in sperm counts, we saw effects in their sons and effects in their grandsons. We said, well, let's run an experiment that's a little more real life. If we could vote this stuff off of the planet, how many generations would it take for things to return to normal? So we took these exposed males and we looked at their sons, we looked at their grandsons, we looked at their great-grandsons, and we looked at their great-great-grandsons. The answer is 3 generations, you're almost back to normal. 4 generations, you are normal for this effect that we study. That's great if you're a mouse. Wonder it's coming. So the problem is, we not only have this bisphenol A, we now have all these replacement bisphenols. And as we were writing this grant saying, we're going to look in our human samples for BPA and the 9 replacements that we think are common, one of the people at our funding agency said, Pat, there's probably more like 50. And my chemist said, wow, we need to not only look for what we know we should be looking for, we need to make a non-targeted screen that allows us to see the things we don't know we should be looking for. Because it isn't just bisphenols. The phthalates are also plasticizers. They also show us effects. And so we have devised a system where we not only look for what we know, but we look for what we don't even know we should be looking for. But it raises huge problems because to me, the biggest thing we need to worry about, well, one of the biggest things, is that the way we regulate these chemicals is we make the feds tell us what's dangerous. They use these old-fashioned studies, hundreds and hundreds of rats, multi-generations, taking organs out and weighing them and looking at them and asking whether they look normal. The problem is these chemicals that behave like hormones, these tests aren't really very good for these types of chemicals. So we need some new tricks. We need better ways of actually asking, what are we being exposed to? All we can really do is take your blood, take your urine, and tell you what's in it, but we can't tell you what you're exposed to. We need better ways of doing that. We need more rapid screens. We can't begin to keep pace with all the chemicals that we are being exposed to. So we need some way that we can do this more rapidly. And biggest one of all, we look chemical by chemical by chemical, and we assume that they're additive. But in fact, I don't know, have you ever gone out on a Friday night and instead of just having beer, you've had beer and then a glass of wine and why not a martini? Yeah, mixtures are kind of worse, aren't they? And there's some evidence from that, from studies, that you can't assume that these are additive. Okay, so how do we do this? We have a challenge system already. How on earth do we do this? I don't know, but I can tell you what I'm thinking. When you make an egg, it doesn't matter if you're a monkey or a mouse or a worm. We see the same exposure effect, whether it's phthalates, whether it's BPA, whether it's replacement bisphenols. What if we use that worm? We can grow thousands of them, thousands, thousands, thousands. We could expose them to everything. We could expose them to every mixture you could imagine. So this is what we're thinking. We're thinking, okay, nobody believes the worm is going to, like, if you go to the feds and say, hey, I got a new plan. We're going to use worms. And this is our tox testing. You're going to get laughed out of the room. So what we're doing is we're partnering together so that our worm colleagues can tell us, here's the ones you should be most concerned about. And we can go back with a furry creature and ask, do we see exactly the same thing, and actually validate it. So we have a lot of work left to do. But one of the things that I think we need to think about is what we as people should be asking for. And I know we need to ask for lots of things. One of the things I think we need to ask for is we really need our regulatory system upgraded. We really need changes to how we regulate the chemicals that come into our life. Better living through chemistry? You betcha. Who could go back to the days before plastics? On the other hand, we not only need to figure out which ones are safer, we need to figure out which ones we're being exposed to and what those exposures actually do in our bodies. It's not just how they degrade, it's how they act and interact in our bodies. All right, I've tried really hard to leave time for questions, and hopefully I have. When I say we, I really mean they, the people in my laboratory who have changed over the years. But additionally, we've had a whole host of colleagues. This is a tight-knit community because we've all been caught out and didn't intend to study these things. But my friends Fred Vomsal and Kathy Vandevert from Davis have been involved in all these monkey studies that I never wanted to do. We have a colleague at Penn who's wonderful in leading us where we need to go in asking why these things affect multiple generations. We have this terrific chemist, Roy Jerome at UCSF, who's been fabulous in helping us not only figure out what chemicals we're being exposed to, but how we should be conducting our human studies. And my worm colleague at Harvard, we think worms are the future.
Are you ready for questions? I imagine there's a few.
I'm ready.
I see a hand way over there.
My question is about how did you administer the BPA to these the mice and the monkeys? Was it through the skin? Was it through a liquid diet?
Yeah, so this is actually really, I don't know if any of you have heard about this big Clarity study where our government put millions of dollars into BPA research. And one of the things they did is they gavaged it, which is they stuffed a tube down the throat and delivered it to the stomach, which is very stressful. We deliver it kind of like a Mars bar. We give them a little in oil, in corn oil. And the first day they fight us, and the second day and third day, and they're looking for their little dose of corn oil. For the monkeys, we put it into fruit. But we wanted to know how it would be metabolized if it went through the skin. Mm-hmm. And we put little silastic implants to actually allow it to deliver like it would through the skin, just slowly.
And that's how you found that the skin delivery was so much more.
So we and several other people, there've been studies looking at, well, estrogen is delivered through the skin, right, for hormone replacement therapy. That's the patch is one way to deliver. So it's a very effective way of delivering estrogens. But yeah, this is what we decided to do. Several other people have done similar studies trying to look at how long it remains bioactive. And it's a difference in what you eat too. So if you eat something watery like soup, goes down fast, gets metabolized quickly. Sure. Peanut butter, it gives a chance for it to go into your bloodstream, and it stays bioactive longer.
So have peanut butter soup, not peanut butter. Thank you. I have a bunch more questions.
Thanks.
Let's go over here to my co-coordinator.
Why not ask, and then there's someone in the back you could take it to. That was terrific, thank you, and to all the speakers. Could you maybe say a little bit about why BPA is even in these products in the first place? Why is it in plastic? Why is it in thermal receipts? What is the, why is it even there? What's the benefit of it? And do you have any comments on maybe just other additives that you have at least glancingly kind of looked at in terms of other chemicals of concern?
Whoa.
Broadly,
okay,
not in detail.
I need 40 more minutes
I know,
now.
I know, sorry.
Alex, you don't mind if we don't go, right?
So when we tumbled upon this first cage, in fact, there happened to be a polymer science department right across the street. And the first thing I did was run over there and say, and they said, we know that there are some harmful effects of this. And I said, well, not like this, you don't. But, I asked all these questions about can't we get a better polymer that's more stable? Because this stuff just breaks down. And what was horrifying to me is, our cages were made of what's called high temperature polycarbonate. I'm not a chemist. I need a chemist. But I don't know what that means. I want garden variety polycarbonate. So I went to the grocery store and I bought baby bottles because at the time, every single one of them was polycarbonate plastic. There were glass ones, yes. I used to give them as presents till they told me they shatter and the daycare won't take them. But we used these baby bottles to try and just dose our mice because we didn't know whether it was coming from our cages, our water bottles. The answer was both. When we touched these baby bottles with the plastic, they fell apart and I freaked out. And so when I talked to reporters, they would always ask, so is there anything else we should have asked her that you want to tell us? And I would say, well, we're putting this into the hands of our most vulnerable segment, our babies and our infants. And I'm really worried about the fact that all these baby bottles and sippy cups are made from polycarbonate plastic. About a month later, I got a call from a baby bottle manufacturer who said, what the hell is going on there? We're getting all these calls from consumers. And then boom, 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 all these new plastics came on the market. And it was like an awakening. Wow, that's how the world works. You tell consumers what to ask for, and then they ask for what they want, and they get it. And that's the way we're going to change things. What was the other half of your question? Other endocrine disruptors. So, the phthalates and the bisphenols are what we think of in terms of plastics. I'm concerned about a lot of things for pesticides and the kinds of disinfectants we use. And again, some new disinfectants are coming on the market. What do you call that stuff that we used to use to sanitize? No. Well, triclosan and triclocarpin hopefully are gone. I always think of peroxide because peroxide is a sanitizer, and some of these new cleaners are peroxide-based, so they just turn into water. So, we can do some really, really amazing things. But in terms of our mice, we have stumbled on a whole bunch of effects. One was a disinfectant that just remains in the environment and just builds up and starts to cause reproductive problems for us. So.
Our next question is up here in the box seats.
Okay, so I'm thinking about products. So one thing, when you're talking about the receipts, I was thinking, receipts. If people are working in that, like if we're just a consumer, maybe we don't get exposed that much. But if somebody day after day is handling the receipts and they're not wearing gloves and maybe they're pregnant, so that was one. And phthalates and shower curtains is the other I was wondering about. And the other thing is, like BPB or the other BPs, like should we be avoiding plastic water bottles, as much as we possibly can? Those are my 3 questions.
I'm never popular. There's almost always a plastic water bottle when I talk to the students, so I harp on them and usually say, why are you still using that? Because it's not BPA free, that's other bisphenols. So that's my harp. Receipts, I can't remember what your first question was about the receipts. What? Oh, somebody who's occupationally exposed. Yeah, it's really interesting. There have been a couple of studies done looking at these kind of individuals. Every time I get on a plane, I look at those people who take your ticket, scan it, hand it back, and do that all day long. If you take one of those people in, you screen their blood before or urine before their shift, in the middle of their shift, and at the end of their shift, you will see the levels go straight up. So yes, it does concern me. There are some really exposed individuals, which is why I think we have some really exposed pregnancies. Time. Thank you.
I'm sorry we don't have time for more questions.